Okay students, so good afternoon to a new lecture. This will be the second lecture of my series that is going on, that is Introduction to Microbial Diversity. Whoever is interested, they can go through this lecture. Already the first lecture has been posted and this is the second lecture. So without any further ado, let's get started. So in my previous lecture, I had already told you about what is classification and why classification was needed, and these were described. Okay, now I told you that in my second lecture I will be starting with principles of biological classification, as this is a bit huge topic, so I'm dividing this topic into bits, because total, if I complete it in a complete video, that will be about 4 hours or 5 hours, so I'm dividing this into 15 to 20 minutes of each video. So if I go into the principles of biological classification, why do we need the principles? Like, why do we need classification system to be coming into play? So classification basically means arranging the living organisms. We are living organisms. So everything, think of a store. In a store also, everything needs to be classified. So similar to that, whenever we are talking about classification, that needs to be classified. Okay, so living organisms needs to be classified into groups, obviously into groups, so that they can be studied more easily. Whatever things we see in our everyday life, everything needs to be in a classified way. Okay, classification not only means that living organisms should be classified, even a store, even a grocery store needs to be classified. How? A section should be made where only roti items will be kept, like flour, atta, maida, whatever. A section should be there where different kinds of rice should be kept. For example, basmati rice, then normal cooking rice, like normal everyday rice which we eat. Then there are different classes of classification. So everything needs to be classified. Okay, so for that reason, scientists basically follow some basic principles to make sure that classification is done in a systematic and universal way. If a store is managed in a systematic way, then that store will be a hit. Like, everyone will think of going to that store. Obviously, if you think of going to a market, you will be always thinking, okay, let's go to so and so store, because in that store you can easily find whatever you want. Okay, you can find easily whatever you want. You don't have to hover around different things and hover around the whole store to get a particular item. You can easily know that, okay, this item is over here in this shelf, so I can get this. I need oil, I can get it from here. I need flour, I can get it from here. So same goes for the scientists as well. They thought that if we don't classify the organisms, then it would be a hectic task for us, for ourselves only, to in our future days it will be difficult for us. For that reason, they basically classified. And why we are talking about biological classification? Because obviously it's organisms, so biological classification. So they classified it based on some shared characteristic features. Okay, organisms are grouped together based on similar features. Obviously, flour will be kept in one shelf, rice will be kept in one shelf, depending on the similar features. Okay, example, think of a bacteria which lacks a true nucleus. They don't have a true nucleus, right? They are prokaryotes, so they are grouped as prokaryotes. Okay, for example, think of fungi. They have cell wall, and that cell wall is made up of chitin. Okay, they are grouped under fungi. So fungi is grouped as a different kingdom. Fungi is a different kingdom. And if we talk about prokaryotes, bacteria, that is a different kingdom. We will talk in our upcoming videos where we will talk about Monera, where the bacteria is basically kept. So the key idea, what was the basic key idea? Why did scientists do? Because they thought that similar traits equals to same group. Similar features equals to let's keep them in the same group. Okay. So the principle of biological classification, basically, see, it's not that much difficult to understand. It's really very, very easy to understand if you go in a proper manner. So my video agenda is to make you people understand in the easiest way possible. Okay, so they thought of making a hierarchy. In our family also we have hierarchy like generation to generation. We are thinking of generation to generation, like we, then our parents, like my father, my father's father, that my grandfather, my great grandfather. That's a hierarchy. So in case of biological classification also, they thought of this hierarchy. So what did they do? First was the kingdom, then was the phylum, class, order, family, genus, and last but not the least, species. Okay, so this classification is stepwise. Now here they have given a very good example, like for kingdom, it's country. Phylum, think of it as a state. Class, think of it as a district. Genus, think of it as a street. Species as the house number. Okay, so for example, think of Escherichia coli, which is the most model organism which we will talk about, as well as Bacillus subtilis. So here, think of Escherichia coli. Kingdom will be what? Bacteria. Phylum will be what? Proteobacteria. Class will be Gamma proteobacteria. Order will be Enterobacteriaceae. Family will be Enterobacteriaceae. Genus will be Escherichia and species will be coli. Okay, so the key idea is from big groups like kingdom to very specific groups like species. Okay, so another thing they focused on was the binomial nomenclature. What is binomial nomenclature? We know what is binomial nomenclature. If anyone doesn't know, they will know in this slide. That is a two-part scientific naming system, that is genus and species. Who was the founder of this binomial nomenclature? Carl Linnaeus. Please ignore if the spelling is wrong. Basically Carl Linnaeus was behind founding this binomial nomenclature classification system. So we will also talk about that in our upcoming slides. So every organism has a two-part scientific name. Okay, so genus is basically the capitalized version and species is the lowercase. So Homo is the genus and sapiens is the species. So how do we write? We write the total in the italicized, like italics. We write it in italics and we underline them as separate words. Okay, we don't underline them together. We underline them as separate genus and species. So names are italicized and underlined. Okay, so this basically what they do, they basically avoid the confusion of local common names. They have given an example. What? Tiger is an English name versus Bagh is a Hindi name. We tell Bagh in Hindi or Bagh in Bengali as well. So both are basically what? Both are Panthera tigris, but because of the common name there can be a confusion. So scientists thought that let's give them a scientific name so that no confusion will be there. So this is a universal naming system. Okay, then came the natural relationship, which is the evolutionary basis. Now what is evolutionary basis? You will tell me, ma'am, it's very difficult to understand, the term is very difficult, so please simplify the term for me. Okay, it tells us how closely organisms are related to each other based on their common ancestors. Based on their common ancestors, how are they related to each other? This is the simple logic and simple definition behind evolutionary relationship. Evolution, how a particular person has, not a person, how a particular how a particular class has how they have evolution, like evolutionary basis, like how have they evolved. Okay, how have they evolved? Our ancestors have evolved and we today we are there, and till now the evolution will go on. Our children, their children, this evolutionary will continue to go on, but they will be depending on the common ancestors. Okay, so modern classification basically considers the evolutionary history of organisms. Organisms that share a more recent common ancestors are considered more closely related. For example, human and chimpanzees, they share a much more common ancestors. Then humans and bacteria. Obviously, humans and bacteria are far, far apart from each other. So they cannot share common ancestral relationship. But human and chimpanzees, they share a common ancestralship. Okay. So classification should show who is related to whom. Okay. Now, universality. Universality means the system must be accepted worldwide. You can't just go and give any system. You can't just go and just think that, okay, let's come up with a new system. It's not like that. You have to come up with a system, or scientists have come up with a system which has been accepted worldwide, so that all scientists can understand. Suddenly you woke up one day and you tell that, okay, I will come up with a new classification system. That would not be accepted worldwide, because that should have some reasons, logic behind the acceptance. Okay, that's why Latin is used as a language, because that Latin is a dead language basically. Nowadays, Latin language is considered as a dead language. That doesn't change. Okay, so that is the reason why Latin languages is used during classification system. For example, bacterium called Staphylococcus aureus will be recognized by this name in every country. Whichever country you go, this name will be recognized. So the key idea was same language for all scientists. We have to come up with a system which will be used by all scientists. Maybe any caste, any nationality they belong, Korean, Chinese, Japanese, Asian, European, whatever. They will know Staphylococcus aureus is the accepted name. Okay. Now we come up with early classification. This is the most important part where we will deal with the early classification attempts before jumping into Whittaker. Because the 5-kingdom classification system, did not come in one day. It did not come up in one day. There were several other classification systems that took place. After that, Whittaker's 5-kingdom classification system came into play. So first one to attempt this was Aristotle in the year of 384 to 322 BC. So this was the first attempt at classification. Okay, he divided the classification group into two: plants and animals. So plants, basically he thought that plants basically meant did not move, got nutrition from soil, and animals were, they could move and they ate food. So very simple, there were no place for microorganisms. They were unknown then, because that time microorganism did not came into play. At the year of 1674, I think so, if I'm not wrong, the microorganisms were known by Anton van Leeuwenhoek, and the limitations was it was too broad that many organisms didn't fit well. Okay, so this was obviously banned. This was it was not banned. It became very old, and it did not play into role later on. Then came the system of Carl Linnaeus. Okay, Carolus Linnaeus or Carl Linnaeus, father of taxonomy, branch of biology that deals with identifying, naming and classifying organisms. So what is taxonomy? Taxonomy is a branch of biology that deals with identifying, naming and classifying organisms. So he introduced the binomial nomenclature system, that is genus and species concept. What he did was that he proposed two kingdoms. One was plantae, one was animae. Plantae, what he did was that all plants were grouped under that, some algae were grouped under that, fungi, bacteria, and animals only contained all animals and protozoa. But there was a limitation. What was the limitation? Many unrelated organisms were placed together. Example, bacteria and plants. Bacteria and plants were placed under that. So obviously bacteria and plants can't be placed under a single rooftop, because bacteria are prokaryotes and plants are what? Plants are eukaryotes. Okay, so they can't be placed under that. So now you will tell me, ma'am, what's the difference between Aristotle and Linnaeus system? Both plants and animals, both have done plants and animals, both have divided into plants and animals. So this was basically 4th century BC, this was 18th century BC. This mainly classified into animals, and this mainly classified both into plants and animals, okay? Used characteristics such as habitat, movement, body, structure, blood, and he used mainly observable morphological characteristic, especially structure of plants, okay? Divided animals into blooded and bloodless. We will come into this. Developed a formal hierarchical classification system. He did not establish the modern scientific naming, but he did binomial nomenclature. His system was an early attempt at organizing organisms, okay? And his system became the foundation of modern taxonomy, okay? Now, what is blooded and what is this bloodless animals means? Blooded animals is basically called as Enaima, and bloodless animals were called as Anaima. Enaima and Anaima, okay? Now, what does this bloodless term mean? Bloodless animals were basically animals that Aristotle believed that did not have the red blood. Like if we cut the animals, blood will not ooze out like we cut human body, okay? In human body, if we cut out, basically blood oozes out. But for example, think of this bloodless group, like for example, worms, then insects, then octopus, mollusks. They basically do not contain a huge amount of blood. So they were, according to him, he classified them as bloodless. And for blooded animals, what he did was the fish, amphibians, reptiles, birds and mammals, okay? This was basically, he grouped them into blooded. Then who came into play? Ernst Haeckel. Ernst Haeckel, in the year of 1866, what he did was that he introduced a new kingdom that is Protista. A new kingdom that is Protista. What does Protista consist? They consist of unicellular organisms such as protozoa, algae, bacteria. So Plantae and Animalia was there, and he introduced Protista. But then there was still a limitation, that is, it was still grouped bacteria with eukaryotes. Like here bacteria was prokaryotes, but protozoa, algae, these were eukaryotes. So again bacteria was clubbed with eukaryotes. So then again a problem came. Then came Copeland. Copeland, what he did was that he proposed a 4-kingdom system, now from 2 to 3, then from 3 to 4. What he did was that he added a new system called Monera. What Monera did was that all prokaryotes, that is bacteria, cyanobacteria, so basically 4 kingdoms, that is Monera, Protista, Plantae, Animalia, okay? Now came Whittaker. What he did was that he introduced this famous 5-kingdom system, where Monera was there, Protista was there, fungi was there, Plantae, Animalia, okay? Based on cell type, like is it prokaryotic, is it eukaryotic? Based on cell number, is it unicellular, is it multicellular? Based on nutrition mode, either it is autotrophic, heterotrophic, and it was a huge improvement. For why? Because accepted worldwide until Oozer system came into play. Okay. Then we will come into binomial nomenclature, okay? Before going into the details of Whittaker, we will just quickly go into binomial nomenclature. What is binomial nomenclature? I have told you what is binomial nomenclature. So basically, binomial nomenclature is a formal system of naming the species, okay? Binomial nomenclature is the So basically this was the early classification attempts, okay? So till now I think so you understood how this early classification attempt till Whittaker they reached, from starting from Aristotle, then Carolus Linnaeus, then Ernst Haeckel, then Copeland, and finally Whittaker. Till here the 5-kingdom classification system has been done, and now in the next lecture we will move on into a new concept of binomial nomenclature, what is binomial nomenclature and what are the advantages of binomial nomenclature, we will go into that in details. Okay, so I will keep this video very short and crisp till this much. I hope you like the video. If you do like the video, do give a like to this video, and don't forget to hit the bell icon so that you can get regular updates if you are liking my video, so that you can get a regular update of when I'm uploading the video. So till then, bye bye. I will see you in the next video. Okay, thank you.